Say what? That was the beautiful city of Cologne, Germany, in case you paid attention. All right, so first of all, it's a great, great honor for me to be here. Thank you so much for having me. It's a particularly big honor to be kind of sandwiched between two people whose work I admire as much as I admire Berthe Forstmann's work and Pascal Fries's work. So yeah, if I do say something entirely stupid and outrageous, which can always happen, just to give me the benefit of the doubt and just accept the fact that I'm more nervous than usual, maybe. All right. So usually if you give me 35 minutes to talk, I will do something programmatic where I try to walk you through kind of our research program and give you some sort of a coherent overview of our work in my lab on the neural basis of cognitive flexibility and how people use cognitive and physiological processes to adapt their behavior to changes in the environment rapidly when they have to. So I'd probably start with something like this, where I would try to convince you that, different behaviorally relevant situations like action errors and unexpected action outcomes and even unexpected perceptual events that your behavior has nothing to do with, but they bear on your behavior, all activate some sort of an overlapping a network of brain regions. And what I would then do is I'd probably try to convince you that part of the reason why they overlap is because the function the computation that they do can be really easily represented in kind of canonical, very algorithmically simple, surprise-based computations that are common across all these situations and lead to the activations of these brain networks. And so then when I've hopefully convinced you of that, I would then go and say, okay, part of what's going on after these surprising events is that we have very specific neurophysiological mechanisms for inhibitory control that the brain then deploys after such surprising events to inhibit ongoing behavior and even to inhibit ongoing cognitive processes. So that's kind of the overview. Instead, what I'm going to do today is I'm actually going to zoom in on just one very specific aspect of this, which are these physiological mechanisms underlying inhibitory control. So computer and hands, well, not that much. Anyway, all right. So what is inhibitory control? And actually, I'm only going to be talking about kind of the lower branch even here of the inhibitory control part when we talk about motor inhibition. So what is inhibitory control? Well, inhibitory control is an umbrella term for cognitive processes or psychological processes that use specific neurophysiological mechanisms that allow you to cancel a movement even after the movement has been initiated. So for example, imagine you're about to step into the street and you see a car bearing down. Even if you've already initiated your walk, you can still kind of rapidly stop yourself without completing the movement even though you've already initiated it. So in the laboratory, obviously, we don't shove people in front of moving cars. We do something like this, which is this task that Berthe already mentioned. It's the stop signal task, very, very simple paradigm. You get a fixation cross, then you get a go signal that tells you to make one of two responses usually, in this case, a left or right hand response. So on that trial, you would have to make a left hand response. But then on a subset of trials, usually a fourth or a third, you will get a second signal after the go signal called the stop signal. In this case, the arrow turns red in the case of this task. And on those trials, what you're supposed to do is, or what that stop signal tells you is, hey, that movement that you're about to make, I told you to make, actually try to stop yourself from carrying out that movement. And you can make it easier or harder for people to successfully stop by displaying the stop signal earlier or later into the motor initiation process. And in fact, behavior in this task can really nicely be modeled by kind of an extremely influential model. The race model of the stop signal task goes back to Gordon Logan's seminal work. And really what the proposition here is, and with some adaptations that model still stands today, is that you have two processes, a go process, which works towards movement initiation and then ultimately execution, that is triggered by the go signal, and a stop process that works towards movement cancellation, that is triggered by the stop signal. And so these two processes race each other to completion. Okay, and then on any given trial, the success of stopping is determined by which of these two processes finishes first. So as a cognitive neuroscientist, I'm obviously interested in the neurophysiological, or generally the brain processes underlying going and stopping, and how they potentially race each other, what their signatures are, and whether you can even read out from brain activity underlying both of these processes, whether you're going to be successful or not at stopping yourself. So if you were to start out in the field and you would go and you would say, okay, well, let's look at what's the neurophysiological signature of going and what's the neurophysiological signature of stopping to make that neural comparison, at least in the EEG or LFP or ECOG literature, and particularly in humans, but also non-human animals, you would find something like this. So this would be activity related to the go process. This is called mu or beta band desynchronization. This is not something we discovered in this paper. That this has been known for decades. But the figures really nicely make the point that I want to make here, which is that if you were to look at go process-related activity, you would look for these beta band desynchronizations or power decreases, however you want to think about them, that occur as soon as you get a stimulus that tells you to make a certain response. So we would look over motor cortices, M1, or sensory motor areas in general, and you would expect to find these kind of smooth desynchronizations that start immediately when the go signal appears, and they basically then go on and align really nicely with response time. Okay, and that's what that the point that figure makes here. So that would be the signature for the go process. What about the stop process? Well, again, you look into the literature and you would find Nikki Swan's seminal work on the stop signal task, and you would see that even the stop process is signified in the local field potential by the increases in beta band power. And here in particular, we're no longer talking about these primary sensory motor areas. We're now talking about control-related areas in prefrontal cortex and frontal cortex, specifically the pre-SMA and the right inferior frontal gyrus, where you'd find these increases in beta band activity. And then Nikki also has a paper where she shows that you can measure that activity on the scalp as well. So these beta band power increases following stop signals. All right, so cool, I'm done, right? The problem is solved. We can just go on individual trials and pull out the processes underlying this race. So on a go trial, I would look for this nice desynchronization signature over motor cortices, and then if there's a stop signal, I would go and look in control-related areas for this nice increase in beta power. Well, of course, I'm only 7 minutes into the talk, so there's a lot more to come, because one core problem with that assumption is that on individual trials, beta band activity does not look like the average at all. And that is not a notion that I came up with. This is really, really cool work by Anne Graybeal's lab and then Stephanie Jones's lab at Brown also. And so basically what they've shown is that, while the averages do indeed look like like these steady synchronizations or power increases or conversely decreases when you talk about the go process, the underlying individual trial data looks like this, and it's really marked not by weaker versions of this that will then show up in the average, but it's dominated by these burst-like transient short latency events that are really, really short in latency. They're kind of scattered out in time, and you can actually miss them by averaging the data up if you don't pay attention. And so the key point here is that not only does the average misrepresent the individual trial data, if you were to apply the logic from the average to look for a relationship to behavior, you wouldn't find one. You will only find reliable relationships to behavior if you actually purely count the number of events that happen in a specific task-relevant period. Now, I should say this is all data from non-human animals, and none of this is necessarily movement-related. These are sensory tasks, essentially, and they interpret this in a sensory fashion. But the point still stands, right? So obviously, being someone that is interested in, like I said, the cognitive neuroscience of, movement inhibition and movement initiation, I would then naturally ask myself, well, what about movement-related signatures in the human brain? Is the same thing that is true for non-human animals during sensory tasks true for motor-related tasks in humans? Now, I would like to brag and say, well, I read all these papers because I'm on top of the literature, and I always read what's in the literature, and then I came up with this great idea. But actually, I was made to read that paper because the Dana Foundation asked me for my opinion on the paper, which I hadn't read, and took me a long time to read and understand because of the complexity and the just sheer volume of the great work that Stephanie Jones's group did there. Also, if you look at the date on this article, in which I really didn't say anything, but yeah, it'd be really nice to have a biophysical theory of what beta waves are. It's almost to the day 3 years ago, so that tells you how slowly my brain works. But anyway, so the question now is the same thing true that, is the same thing that Stephanie Jones's group and Anne Graybeal's group find in non-human animals, is the same thing true for these go and stop related signatures in the human EEG? And I'm actually going to give the game away already. This is a figure that I made that is made to look like one of Stephanie Jones's figures. So this is the average of, this is real EEG data from one of our subjects. These are 6 individual trials, and that is the average of those trials. So if you were to look at the average, you would say, okay, on each individual trial, I'll just look for this signature, but indeed what the individual trial signatures look like are much shorter burst-like transient events. But one step after the other. So what did I actually do in the current study? So I basically gathered all the EEG recordings that we had in my lab from stop signal task data sets in healthy young humans. 64-channel EEG. A lot of these data have already been published. We often use the stop signal task as a functional localizer for these inhibitory processes, but a lot of that data is also just de novo data that we kind of keep collecting as we go on in the lab. So now let's go and actually look at, go and stop related responses. So let's first of all look at the average data underlying the go process, and we see exactly what we would expect if we were to look at the average. This is filtered beta, this is beta band power at these sensory motor electrode sites C3 and C4. If you're an EEG person, you will know that that's generally where you would look for motor-related activities like the LRP or these types of beta burst desynchronizations, beta power desynchronizations. So you basically see this nice desynchronization. It's slow and steady and it looks consistent and whatnot. And now let's look at the individual trials, and that is what the underlying individual trial data looks like, right? And so what's really striking here is that, and these are, I should say, entirely randomly selected trials, right? This is a random number generator that pulls from the tens of thousands of trials that we have and just pulls out a couple for each of these electrodes across different subjects. And so you can see that instead of being marked by like a slow and steady desynchronization, you can see these, these transient burst-like events. Now, obviously, that is somewhat qualitative, or it's actually entirely qualitative. If you're looking at it and you say, well, these are beta bursts, it's not, they're not spread out in time. You can quantify, so the alternative would be that you have an ongoing oscillation that's just amplitude modulated kind of transiently instead of it being an actual separate burst-like event. But if it is an ongoing oscillation, and maybe you just can't see it over the noise or something, what you would expect to see is an ongoing phase relationship within the signal itself. So in other words, you should be able to explain the phase of the signal at any given point in time from the previous phase of that same signal at the same electrode on the same trial. This is a method called lagged coherence that's been developed by Eric Morris's lab. And if you apply that method, what you can see is that actually, so this is the lag coherence index here on the y-axis, and then frequency on the x-axis. And you can actually see that all across the beta band, there's actually a trough in the lag coherence. So there are some of these, intertemporal phase relationships in the alpha band, but the beta band seems free from this. Now, this is still, you can still, it's still not definitive, but, between the qualitative judgment of looking at the data and the quantitative evidence of the lag coherence, I think it's somewhat safe to say, especially when you look at Stephanie Jones's and Anne Grable's data as well, that there seems to be something to this burst-like nature of the signal. So now let's look at these bursts a little bit more systematically when it comes to the go process. These are now topographical representations of the signal. If you're an EEG person, you're used to looking at these in terms of amplitude of an ERP or of power or whatever. And I want you to not forget about that, not do that, because what this is it's literally just the count of the number of beta bursts at different time points following a go signal, separately for left-hand responses and right-hand responses. And what you can see by looking at these plots is that at the beginning of the trial, when the go signal hasn't really been even visually computed yet, there seems to be this tonic ongoing beta bursting centered around the two sensory motor electrode sites, C3 and C4. And that as you get closer to response emission, remember these are all go trials, there are no stop signals here, so they are responding on each of these trials, that signature keeps going away. And you can see the same thing in the histograms. So these are the beta burst histograms separately for C4 and C3 now. And so you can see this steady decrease as you get closer to the response. And interestingly enough, that beta burst reduction, the reduction in the number of these beta bursts lateralizes to the contralateral electrode site as you get closer to the response. So really what this indicates is that the feature of the average, which is this, this steady-looking desynchronization, what's underlying that on the individual trial level is actually a steady decrease in bursts that shows the same features essentially. But what these bursts give you is they now, and this is again in line with Stephanie Jones's work in these rodents, they give you a better handle on the relationship between brain activity and behavior. Because here's a correlation that you can only produce if you quantify and look at the data in terms of beta bursts. So what this is, it looks more complicated than it needs to be. It's really just a correlation between the number of beta bursts in each of these time windows for each individual subject over the contralateral motor site. And the correlation, the green dots here, is with go reaction time. And what I want to point you to, and this is obviously corrected from multiple comparisons, et cetera, but what I want to point you to is that around 100 milliseconds, so in the early trial period, the amount of beta bursts at the contralateral sensory motor site predicts the go reaction time, in that participants who have more beta bursts at these sensory motor sites when the go signal appears take longer to initiate a response. So the appealing interpretation, and I will admit obviously that this is an interpretation at this point, is that potentially these tonic sensory motor beta bursts reflect a net inhibited state of the motor system that you have to overcome in order to initiate a response. Okay, so that's the go process. Let's now look at the stop process. So what I'm doing here now is I'm literally just comparing the numbers of beta bursts at each individual scalp site between stop trials and go trials. So these are successful stop trials on top, failed stop trials, so stop trials where there was a stop signal but they responded anyway on the bottom, and subtracted from that is the go beta bursts in matched time periods on matched go trials. And so what you can see here is that there is a spatiotemporal coherence that develops in these beta bursts after stop signals, and everything that, by the way, everything that you can see here that's different from 0 on this scale is significant corrected for multiple comparisons across all time periods, across all 64 electrodes. Remember, we have 234 people here, so there's a lot of power to be had. But anyway, so if you look at this plot, you can see that at these frontocentral electrode sites, kind of, the centroid being at the electrode FCZ, there is, at around 200 milliseconds following the stop signal, this phasic increase in beta bursts, or at least in the number of beta burst events. Now, I do know that I haven't shown you the individual trial data yet, so this is individual trial data at electrode FCZ, right? So this is what is underlying that previous plot. And again, you can see that as far as visually detectable features go, these trials are clearly marked by the occurrence of these transient, really short-lived beta burst events. Whereas the average, again, looks kind of like a smooth change in activity. Now, the question again is, just with the go process here, does this relate to the success of stopping? Does this relate to behavior in systematic ways? And indeed it does. So this plot is pretty simple. It's just a comparison of beta burst count on successful and failed stop trials compared to matched go trials and matched time periods on matched go trials. And you can see two things here. There is an increase, the beta burst increase on successful stop trials is greater compared to both matched go and compared to failed. And the interesting part here is, this is highly significant, obviously, given our large sample. The effect size is, 0.3 something, so it's not huge. But what's important here is, even with our 230 whatever participants, if you looked at the average amplitude, and if you had your single trial analysis informed by what the average looks like, and you just pulled out an average beta power amplitude, you would not find any difference there whatsoever. Okay, so now we have a somewhat potential novel signature of, of motor inhibition. So, of movement initiation, movement cancellation. Let's see if we can actually use that to, speak towards some of the debates in the field. Well, one of the debates that are going on in the field of inhibitory control is the question of the nature of the race that is underlying the stop signal task. So the initial conceptualization by Gordon Logan, like I said, was that you have this independent race model. So you have the two processes that race each other. Whichever one finishes first determines whether you succeed or fail at stopping. There is another alternative here that's been proposed by Jeff Schall's lab. Gordon Logan is also on that paper, but this is basically an interactive race model. And so the difference here is that in that conceptualization, as soon as the stop process the rough conceptualization is as soon as the stop process starts, it starts actively acting on the go process. So it qualitatively changes the go process, potentially in an inhibitory or suppressive fashion. So can the beta bursts tell us something about whether that's the case or not? And they actually can. So this is a more complicated plot than it needs to be. Really all I did here is for each individual stop trial, I looked for the first frontocentral beta burst that occurred after the stop signal, and I time-locked the sensory motor signal to the occurrence of that frontocentral beta burst. And then I asked the question, is there a change in sensory motor beta bursting when there is a frontocentral beta burst, so a phasic frontocentral beta burst after a stop signal, compared to a matched time period where there is not one? And what you can see here is that indeed there is a change in that following the frontocentral beta bursts, the sensory motor beta bursts reinitiate essentially. So on an individual stop trial, basically the way you conceptualize this, at least from the perspective of these local field potentials as they're collected from scalp EEG, you would see the following. You'd get a go signal, so you'd have tonic ongoing sensory motor beta bursting. You'd get a go signal that as you initiate the movement, the beta bursting would reduce over motor areas. Then you would get a phasic increase when there is a stop signal. You would get a phasic increase of beta bursting over frontocentral sites. And then immediately after that, within 25 to 50 milliseconds after that, the sensory motor beta bursting would go up again. And so what that speaks towards is really this interactive nature of this race. At least the suggestion here, some concerns about volume conduction that I can gladly address here notwithstanding, what you could take from this is to say that indeed the race seems to be interactive in that when the stop process occurs, if the stop process indeed is signaled by these phasic beta bursts, it does change the go process in that it reinitiates this initial state of inhibition. All right, so let me give you a preliminary summary here. So, both lateral sensory motor beta during movement initiation and frontocentral beta during movement cancellation is burst-like in nature on individual trials. These bursts relate to behavior in systematic ways above and beyond what you could glean from an average quantification. And burst-locked analyses, like the last one that I just showed to you, allow novel insights into the potential interactions between these neurophysiological mechanisms underlying going and stopping. Okay, so that's the first part of my talk. In the next 10 or 15 minutes that I still have, let me zoom back out. And I told you I was going to get back to surprise at some point. So I'm doing that now. So for now, all I talked about was motor inhibition, but what I already told you is that a lot of the work in our lab and a lot of my own past work really relates to this fact that motor inhibition or these physiologically inhibitory processes become automatically activated following surprising events. So in other words, even after events where you're not explicitly told, hey, stop the action you're about to make, you are inhibiting your ongoing movement. And so, we've shown that in different, imaging domains, including, some SCN data that refers to some of what Berta presented. And I'll give you a couple of examples of how we've shown that. But obviously now the question is, well, if surprising events trigger inhibition, which I'll hopefully convince you of in the next 5 or 10 minutes, is there a phasic increase in frontocentral beta bursts, if that indeed is the signature of motor inhibition? Okay. So let me very briefly tell you about why I think that surprising events, even though surprising events don't tell you anything about having to stop your movement. And actually in the cognitive neuroscience science literature for decades, we talk about surprise, obviously, but we talk about surprise in very cognitive terms, like model updating or attentional orienting, or we talk about the autonomic nervous system. And so I think one part that's underappreciated is that part of the brain's automatic response to surprising events is to actually stop motor activity or momentarily inhibit motor activity. And that actually makes a lot of sense if you think about surprising events in the real world. When you're doing something, but your environment behaves in a way that you didn't predict when you were planning and executing the movement, you do want to stop a little bit and think about whether what you're doing is still appropriate given the unexpected environmental demands. Okay, so to me the most straightforward evidence for the fact that surprising events induce motor inhibition comes from the actual physiological excitability of the motor system itself, which you can measure using transcranial magnetic stimulation. It's a really simple method. You put an EMG lead on a target muscle that you're interested in, for example, a hand muscle, and then you stimulate the contralateral motor representation of that hand muscle with a single pulse of TMS. And what that does is it produces a deflection in the EMG trace from the target muscle that's called the motor evoked potential. And the amplitude of that motor evoked potential is a direct proxy for the net corticospinal excitability of that specific motor tract. So if you were to do something like that during the stop signal task, what you would find is that when people successfully stop a movement, you would find suppression of that cortical motor tract, and you would actually find a reduction of the MEP even below the resting baseline, which really nicely suggests that there is some active inhibition of the motor system going on. The interesting part about the stop signal task is that this inhibition, and this is a feature that I find really neat about it, this inhibition is non-selective or global. In other words, what that means is, regardless of which motor effector you stop, you will find suppression even of task-unrelated motor effectors. So for example, in this particular study, we had participants make eye movements and stop saccades to the left or to the right-hand side, and their hands were resting on the table, they were not using their hands at all. But what we found is that when participants successfully stopped their eye movements, there was a suppression of the task-unrelated hand, so of the motor excitability of the task-unrelated hand. We found the same, and other groups have found this too now, that even with different combinations of motor effectors, like you stop your hand but you measure the MEP at the leg, or you stop even your voice and you measure the MEP at the task-unrelated hand again, you will always find a suppression of the motor evoked potential following even in task-unrelated effectors. Now, before I get to surprise and show you that the same thing happens after surprise, just to kind of spoil the surprise, haha, I want to briefly bring back this plot. If you've been paying attention to this admittedly convoluted plot, what you can see here is that the reinitiation of sensory motor beta bursting following frontocentral beta bursts during stopping is bilateral. So it's not specific to the movement that you're actually stopping. The beta bursting doesn't just go up over left motor cortex when you're making a right-hand movement. It goes up on both sides. And I think that might be the neurophysiological correlate of this non-selective inhibition that you can then observe at the level of the motor system. But that's just a side note. Now let me finally get to surprise, like I promised, actually. So let's just actually investigate whether there is non-selective suppression of the motor system during surprising events. Very, very simple task. This is a very simple reaction time task. You get a fixation cross. At some point, you see a letter on the screen. You have to say the letter, so it's a vocal response. But 500 milliseconds before the imperative stimulus, you get a cue. And the participants practice with the same sound cue over and over again. It's always the same sine wave tone, except on the actual task, we then swap in these unexpected birdsong segments on a subset of trials. All the while, we're measuring motor excitability from the task-unrelated hand muscle that's resting on the table. And so what we can find here, first of all, is a reaction time effect. So when there's an unexpected cue prior to the imperative stimulus, you see a reaction time slowing to the imperative stimulus. But even more importantly, what you can see is you actually see a suppression of the motor evoked potential on unexpected sounds compared to expected sounds, and importantly, even compared to baseline. And we've also replicated this now in a much larger sample. So, and this is specifically actually at 150 milliseconds following the unexpected cue. Okay, so just to really drive the point home that surprising events actually automatically invoke motor inhibition, I want to show you a brief behavioral experiment here. What I basically did here is I paired this is a motor inhibition task. It's a go-no-go task, which is not an ideal version of a motor inhibition task. This was a modification of an existing study. That's why I went for a go-no-go task. If you design a go-no-go task properly with a low percentage of no-go trials and a very fast response window, it becomes a motor inhibition task, and you can actually find prepotent motor activity on successful no-go trials even. But the point here is that we paired the no-go and the go stimulus with these sounds again and had participants practice with the same sound over and over and over again. And then we swapped out the sound for an unexpected sound on some trials in the actual experiment. Now notice the two contrary predictions that you would make here depending on what perspective you apply. From a motor inhibition perspective, if you believe that surprising events activate the motor inhibition system, they should help you successfully stop. From a distraction perspective, from a classic cognitive surprise perspective, you would imagine that you could get worse at the task. Their failed stop rates will go up when there's a surprising tone because it pulls their attention away from the primary task. But that's not the case, otherwise I wouldn't be talking about it, I suppose. So these are about 88 subjects or something doing this task, and you can see that clearly the probability of making a failed stop or of responding when there's a stop signal is much lower when the no-go signal is paired with an unexpected tone. And just briefly, what Isabella Dutra, who's an undergraduate in my lab, and Darcy Waller, who's a graduate student in my lab, did is they then used this task and applied our TMS logic to it. So they had participants do this task with their feet, which makes their hands unrelated, task unrelated, hands are resting on the table. And so what they then did is they basically asked the question, well, does the expected non-selective suppression of the task unrelated hand muscle following a no-go signal become amplified by an unexpected event, and moreover, is that related to the behavioral benefit that they incur? And long story short, they basically found both. So in other words, there is a suppression, a general suppression of no-go versus go. That is just our non-selective suppression during motor inhibition, but that is amplified by the additional presence of the unexpected tone. And moreover, the degree of behavioral benefit that is incurred on individual subjects is directly related to the degree of additional physiological inhibition that is brought about by the presence of the unexpected tone. Okay, so now let's come back to beta bursts. So this is all previously existing work on the tight link between the occurrence of surprising events, even outside of the context of an explicit motor inhibition task, that automatically invoke motor inhibition. So that would make the prediction if, based on our stop signal data set, where I said that a phasic increase in frontocentral beta bursts would signify the presence of this inhibition process, you should find the same thing following surprising events outside of a stopping context even. So this is a reanalysis of these data. This paper is not about beta bursts, but it uses the same very simple reaction time task with the expected and unexpected cues that I talked about earlier. And so what we can do here then, this is 55 participants, and we can look at frontocentral beta bursts following unexpected cues compared to expected cues. And what you can see here really nicely is that there is a really nice increase of frontocentral beta bursts at around 150 milliseconds following the unexpected event. Now, remember what I said about the timing of the non-selective suppression of the motor system in the TMS method. That occurs at the exact same time point. And then finally, and this is a plot I made last night, so it's not super pretty, but this is basically the same type of analysis. Oh, this is wrong. This is not supposed to say tone. This is supposed to say frontocentral beta burst following an unexpected tone. And then what you can see here is the sensory motor beta bursts at different time points following that frontocentral beta burst. And you can see that these go up. And that is all in line with this behavioral effect of reaction time slowing that you find following these unexpected events. Okay, so let me sum this up. Surprising events automatically incur motor inhibition. That inhibition is accompanied by a phasic increase in frontocentral beta bursts. That increase in frontocentral beta is followed by an immediate increase in sensory motor beta bursting, just as it was for the explicit stop signal task. So my tentative interpretation is that tonic sensory motor beta bursting reflects a net inhibited state of the motor system. That state has to be overcome during movement initiation, and then phasic increases in frontocentral beta bursts can lead to a momentary reinstantiation of inhibition. These are the people in my lab. This picture has been taken 1 week ago in my front yard, just to make the point that the weather in Iowa is actually much nicer than the weather in Germany, so please stop asking me. The winters are bad, it's true. But yes, thank you very much for your attention. Thanks for having me here.
Great. Thanks, Jan.